Cell biology in the context of medical science is a fascinating and ever-evolving field. From the intricate processes that occur within cells to the complex interplay between different cell types, a deep understanding of cell biology is crucial in diagnosing and treating various diseases. In this detailed script, we will explore 12 different topics related to cell biology in the context of lung function and disease. We will delve into the nuances and mechanisms that contribute to the normal functioning of the lung and how abnormalities in cellular processes can lead to respiratory disorders. So, let's begin our journey into the intricate world of cell biology in the lungs. Alveolar epithelial cells are important for gas exchange in the lungs. They differentiate through specific factors and pathways. One key factor is NKX2-1, which promotes surfactant production and gas exchange. WNT/beta-catenin signaling regulates proliferation and differentiation. This understanding is essential for treating lung diseases. Surfactant, made by lung cells, reduces tension in alveoli, preventing collapse. Surfactant protein D helps clear pathogens. Hormones regulate secretion. Lipid synthesis and protein production reduce tension. Problems lead to respiratory distress in newborns and lung injury in adults. Enhanced production may help patients with surfactant disorders. Epithelial mesenchymal transition, EMT, is a cellular process where lung cells change their characteristics, which is important for lung development. However, when this process goes wrong, it can cause lung fibrosis and cancer. Understanding EMT is key to developing treatments for these conditions. Ion channels are proteins that control the movement of ions across cell membranes. In pulmonary cells, ion channels are important for maintaining balance and regulating membrane potential. The cystic fibrosis transmembrane conductance regulator, CFTR, is a well-known ion channel responsible for regulating chloride and bicarbonate transport. Dysfunction of CFTR leads to cystic fibrosis. Other ion channels, like ANAC and aquaporins, are crucial for maintaining lung homeostasis. Studying these channels helps understand respiratory disorders and develop treatments. Pulmonary neuroendocrine cells, NCs, are specialized cells in the airway that regulate breathing and immune responses. They sense changes in the air and release chemicals to control muscle tone, mucus production, and immune response. Problems with NCs contribute to asthma and COPD, and studying them may lead to new treatments. In lung fibrosis, excessive accumulation of extracellular matrix proteins distorts lung architecture. Epithelial cell injury releases profibrotic factors, triggering fibroblast activation and myofibroblast formation. Immune cells release pro-inflammatory mediators, perpetuating fibrosis. Understanding these mechanisms is crucial for developing targeted therapies. Pulmonary vascular remodeling refers to the structural changes that occur in the pulmonary blood vessels, leading to increased vascular resistance and impaired blood flow. This process is a hallmark of pulmonary hypertension, a progressive and life-threatening disorder. Several signaling pathways are involved in the regulation of pulmonary vascular remodeling, including the transforming growth factor beta, TGF-beta, endothelin-1, ET1, and prostacyclin pathways. Dysregulation of these pathways leads to excessive proliferation of smooth muscle cells, endothelial dysfunction, and vascular remodeling. Understanding the molecular mechanisms underlying pulmonary vascular remodeling is crucial for the development of targeted therapies that can specifically modulate these pathways and improve blood flow in patients with pulmonary hypertension. Alveolar macrophages are immune cells in the lungs that help maintain lung health. They engulf harmful microorganisms, clear debris, and release inflammatory substances. They interact with other immune cells to shape immune responses. Dysregulation of their function can lead to lung infections and inflammation. Stem/progenitor cells have been identified in various compartments of the lung, including the bronchiolar epithelium, alveolar epithelium, and vasculature. These cells possess the capacity to differentiate into specialized cell types and contribute to tissue repair and regeneration. In response to lung injury, resident stem/progenitor cells can be activated and undergo proliferation and differentiation to replace damaged cells. Additionally, bone marrow-derived stem cells can migrate to the injured lung and participate in tissue repair. The identification and characterization of lung stem/progenitor cells hold promising implications for developing regenerative therapies to treat chronic lung diseases, including COPD and IPF. Understanding the precise mechanisms that regulate the activation, recruitment, and differentiation of these cells is crucial for harnessing their regenerative potential. Pulmonary inflammation is a key aspect of asthma, a chronic respiratory disease marked by recurrent airway narrowing, coughing, and wheezing. It involves communication between immune and structural cells, which release inflammatory substances causing bronchoconstriction, mucus production, and immune cell infiltration. This knowledge is vital for developing effective anti-inflammatory treatments. Pulmonary neuroepithelial bodies, PNEBs, are specialized cells in the airway epithelium involved in chemoreception of oxygen and carbon dioxide levels. Dysfunction of PNEBs is linked to respiratory disorders. Studying function may lead to new diagnostic and treatment options for these conditions. Autophagy is a cellular process involved in the recycling and degradation of damaged organelles and proteins. It plays a crucial role in maintaining cellular homeostasis, eliminating toxic aggregates, and responding to stress. In the context of lung health and disease, autophagy has been implicated in various processes, including alveolar epithelial cell remodeling, surfactant homeostasis, and lung infections. Dysregulation of autophagy has been associated with lung diseases such as COPD, pulmonary fibrosis, and lung cancer. Understanding the intricate molecular mechanisms underlying autophagy in the lungs can provide insights into the pathogenesis of these disorders and offer potential therapeutic targets for intervention. In summary, the respiratory system relies on intricate cellular processes, such as alveolar cell differentiation and stem cell involvement, to maintain lung function. Exploring topics like vascular remodeling, neuroendocrine cells, and autophagy can enhance our understanding of respiratory regulation and disease. Studying these cellular mechanisms is crucial for developing effective treatments for respiratory disorders and improving patient outcomes.